My name is Morten Karsdal. I'm head of R&D, CEO at Nordic Bioscience. Collagens. I love collagens. My team and I, we wrote the first collagen book in 2017. I love teamwork. Now we've just made the third edition. And it's a university-grade book, really lots of insights and good collagen research. It's a big question. I think if I could ask a different question, I would say, which collagen are you the most afraid of? And clearly, I am the most afraid of type 6 collagen, because I think that's where the link between wound healing and fibrogenesis actually lies. We do not completely understand it yet, but as I showed earlier, all the patients with elevated levels of endotrophin, they will die. And we do not know it's from cardiac or renal, but clearly in that fibrotic activation, there is a link to both fibrosis and wound healing that is super dangerous. I think that is up for others to really decide. I have been very fortunate to be participating in 7 different approved biomarkers by the FDA and EMA, and each one of them provides something differently. It was my team who originally developed CTX1, the bone resorption biomarker that everybody's using. We've also done ProC3 that's just used in Europe, will be approved in the US. I think they provide something different. And I don't have a favorite poster child here. I just think it's important that we understand that fibroblast activity is super dangerous. And patients that have elevated levels of fibroblast activity, we need to quantify that, we need to monitor that, we need to change that, because there are 40% of patients in the Western world that are dying from fibroactive disorders. And that's really big for me to say that to everybody, because it's about basic research, but it's also about patients and quantifying and changing lives. I'm brought up on doing antibodies. I am a big believer in the specificity of antibodies, and so we only work with monoclonal antibodies. And there's also a lot of bad research on collagens because they are like brother and sister molecules. So if you have a monoclonal antibodies, that means yes, it will react to your collagen and all the other collagens. So what I'm mostly interested in when I look at a technology is the fact, yes, we do positive selection. We make sure that this antibody does bind to this sequence. But then we need to make sure that it does not bind to that sequence. And that is one of the problems with the aptamer technology, with other technologies, that they've done positive selection, not negative selection. So yeah, mainly antibodies that we work with, and then tons of human blood. I'm just disappointed in myself enough. I've been attending earlier. I have been interested in the matrix for a long time. It's the biggest society for matrix biology. So I think I should have been here earlier. I'm enjoying myself. I love the matrix. And I think it's important that we realize that the collagens may be called collagens, but they are a diverse family, super diverse family, and they do very, very different things. So we invited to the Extracellular Matrix Pharmacology Conference, that is a bi-yearly event in Copenhagen, Denmark. We invited a few of the American Society of Matrix Biology participants. They enjoyed their time. Tom invited me back here, and I think the session we had today with Philip Scherer, Philip is, he discovered leptin, he discovered adiponectin, he discovered endotrophin. He's Nobel class material. That's it. That's a big question. Okay. So initially, that there is not a defined ligand. If you think of how you develop drugs, you need a ligand and a target. And then the process is pretty simple. Then you do whatever it is that we do, and there's a path. However, when you figure out that there's a protein in the matrix that may not be targetable, it may not be accessible, and it's big, then what part of that protein is the druggable part? So I think it's very different from a traditional ligand and receptor. Thinking of 7TM receptors are the most druggable targets. It's not super druggable targets, and we don't know if we should be targeting the ligand, removing the ligand, or finding the receptor. I think the most important thing I would say what I actually decided to do when I started. I think it was important to do something that within a foreseeable future could be translated into something that was a benefit for man. Of course, we need basic research, but we also accept that there is a tendency to invest more if there is a potential route to benefit for patients. I would call that applied research. And I think many times that it is not seen as fancy, but that application of tools and technologies to advance something that we can use in medical science, I think that provides an alternative. Also because that would allow that young scientist to have multiple different career paths. There can be academic ones, there can be one in medtech, there can be one in pharma. And so, as I always tell my kids, I would make sure that my next decision is leaving the next many decisions open. And I think choosing applied research allows us to go in many directions. Bench to bedside, honestly, for me, that I've heard that for so long time, and that gets mistranslated into what I think is interesting for something. And I think it's very important to have a clear view that what is the end user, who is actually going to use this, and what is the decision that that person, doctor, caregiver, is going to make based on that tool. And what is the road to that? That was the biomarker valley of death. What is the tools and technologies that we're going to be developing? So I want more granularity into that sentence. We're midway here, and there will be more. But of course, I love being in a setting where matrix is the center of the stage. Originally, we did the extracellular matrix pharmacology conference in Copenhagen, Denmark, because I got a little tired of being in a corner at a liver conference, in the corner in a kidney conference, because I am convinced that if we're going to change people's lives, we need to be changing the matrix of those organs. And so I love the fact that we are all here with an aim to understand and change the matrix. I did love the basement membrane session yesterday, and the Boris Hinz session yesterday on fibrosis and wound healing. I think that was also an amazing session. So there are many good things, and I'm sure there are more to come. I thought I was a cat person, but then it turned out we got a small furry dog. And sometimes that is the best member of the family. At least it is the family member that is welcoming me home the best.